Chapter 6: Lipids, Membranes, and the First Cell. In this chapter, we're going to talk about lipid structure and function, and talk about lipids and how they can spontaneously form, and how they move, and then we'll talk about how substances move across bilayers. In other words, we'll focus on diffusion and osmosis, and then look at membrane proteins that facilitate diffusion. So what makes the plasma membrane or cell membrane that separates a cell from life or non-life? Well, it's made of lipids. It's just like my skin. My skin defines me, and it's the boundary of me, and it separates me from my environment. So a plasma membrane separates the cell interior from the external environment. So what is the exact function of a cell membrane besides this separation? It's to keep damaging materials out and allow needed materials to go into the cell, and it's also used to facilitate chemical reactions. Lipids are made of carbon, and they're found in organisms, and they're largely nonpolar and hydrophobic. So remember what hydrophobic means? It means water hating. And nonpolar means that electrons are shared equally in covalent bonds. So lipids are composed of hydrocarbons, at least many of them are, and this is a carbon backbone with hydrogens coming out from the sides. Hydrocarbons are hydrophobic, and electrons are shared equally between carbon and hydrogen bonds, and also carbon-carbon bonds. So how does bond saturation affect hydrocarbon structure? Well, an isoprenoid is a hydrocarbon. So a hydrocarbon, again, is a long chain of carbons that acts as a backbone, and out from the carbon come hydrogen. And pigments, scents, vitamins, and sex hormones can be made of hydrocarbons. And these hydrocarbons can also build more complex lipids or fats. So an essential structure is the fatty acid. What is a fatty acid? It's a hydrocarbon chain, but it has a carboxyl group at one end. Carboxyl is COOH. And it says here a fatty acid can be saturated or unsaturated. Well, here we have in this diagram 6.1, we first see an isoprenoid, and we see that it's a series of isoprenes connected together in a long chain. But the bottom line is it has a backbone of carbon with hydrogen at its side. So when you see that zigzag pattern, what you should imagine in your mind always is carbon at each of the corners with bonds made to hydrogen. If you look at a saturated fatty acid, which is in the center diagram, you see that the chain is rather long and straight, and there's all single bonds. There's no double bonds, and so this would be a saturated fatty acid. In other words, it's saturated with hydrogen. And then we have an unsaturated fatty acid on the right-hand side. So what's the difference? Well, I see some double bonds. Instead of single bonds, there's some double bonds between carbon and another carbon. That means it's missing some hydrogens. And when you have a double bond, at that point, there's a kink or a bend. And this is important because it changes the density of a lipid. So let's go over this again. So what makes a saturated hydrocarbon saturated? It only has single bonds, and it's saturated with hydrogen. In other words, it has the maximum number of hydrogen atoms. On the other hand, unsaturated hydrocarbon chains have at least one or more double bonds. They are not saturated with hydrogen, and this double bond causes kinks in the chain. So when there's kinks in the chain, then there's more space and it's less dense. So how does bond saturation affect hydrocarbon structure? Well, we know from commercials that polyunsaturated lipids may prevent heart disease. On the other hand, saturated lipids such as butter or maybe bacon fat, they're solid at room temperature because the tails, the fatty acid tails, are long and straight because they're saturated with hydrogen and there's only single bonds. So they become very stiff, they're denser at room temperature, and that's why we see them as solids. So it says here highly unsaturated lipids are liquid at room temperature. And I'm thinking of like corn oil, olive oil, safflower oil. They're all unsaturated because they have double bonds. In other words, they're not saturated with hydrogen. And so therefore they're less dense at room temperature and they present themselves in the liquid form. I love this example, and you could have a test question like what affects the permeability of a membrane? So notice we have butter, beeswax, and safflower oil. Butter and beeswax, they're saturated hydrocarbons, but if you look at the difference between the two, the obvious difference is that beeswax has such a long hydrocarbon chain, which whereas butter is much shorter. So which one is the least permeable to molecules moving across? It's definitely the beeswax, and that's because the hydrocarbon chain is very long. Then in part C we see safflower oil, and notice that there's double bonds. And in fact there's two sets of double bonds indicating kinks and a less dense lipid. So that's why you see a liquid at room temperature. And then lastly at the bottom we see Crisco, and this is made from hydrogenation. That means that in the lab that there's been pressure to actually add hydrogen to the oil to make it a solid. This is very unnatural, and so you should avoid using unnatural fats at all costs. It's better to use natural fats than something that is unnatural. There are three types of lipids found in cells. But regardless of the types, the one thing that we can say about all lipids is that they're insoluble in water. And that sounds like a test question. So instead of looking at the chemical structure, we're looking at the fact that they're all hydrophobic or insoluble in water. So the reason for this is because they're made of mainly nonpolar covalent bonds. The nonpolar covalent bonds are found between carbon-carbon and carbon-hydrogen bonds. So what are the three types of fats? Steroids, fats, and phospholipids. But truly, after we identify these three fats, most of this chapter is going to be about phospholipids. Why? Because phospholipids are the type of lipids that make the plasma membrane for every cell. The first type of lipid are steroids, and steroids have a four-ring structure. So if you're out in the gym and you're exercising, you might be interested in taking steroids to bulk up, or they call it roids at the gym. And I can often see people drinking a lot of water when they're taking steroids because I think it's hard on your kidney. But the truth is that steroids look totally different than phospholipids or triglycerides. But what makes them all the same is that steroids are insoluble in water. So what are some famous steroids? Estrogen, testosterone, and cholesterol. So we'll talk about cholesterol because we find cholesterol in plasma membranes. So this is what a steroid looks like. You see the four-chain structure. And notice this four-chain structure has a hydrophilic and a hydrophobic side. It is both polar and nonpolar, and we'll learn that that word is amphipathic. Amphipathic. The next type of lipids are fats. They're often called triglycerides or triacylglycerols. So sometimes when you have someone in your family and they've done blood work, you'll see that they measure their triglycerides. In other words, the amount of fat flowing through your veins. So when their fatty acids are polyunsaturated, they're liquid and form oils. But the primary role of fats is to store energy. And remember from the previous chapter, each molecule of fat stores more energy than a molecule of sugar. And the reason is because fats are made out of nonpolar covalent bonds. So it says fats form dehydration reactions between a hydroxyl group of a glycerol and the carboxyl group of a fatty acid. And when they do this, this is an ester linkage. This will not be on the test, and it's just something that's important to know for later on. And even though there is a dehydration reaction, this does not imply that fats are polymers. In other words, fats are not polymers at all. So here's the formation of a triglyceride, and just by looking at it you can see where it gets its name. There's 3 fatty acid tails connected to a glycerol molecule. It sort of looks like a jellyfish. So this is a triglyceride. The last group of lipids are called phospholipids, and they too consist of a glycerol, but instead of having 3 fatty acid tails, they only have 2. One has been removed and replaced by a phosphate head, and this is how they get their name, phospholipids. Phospholipids are important because the primary role of phospholipids is to form cell membranes. So this is what a phospholipid looks like. You can see glycerol and 2 fatty acid tails, but the third one has been removed and replaced by a phosphate. In addition to the phosphate, we see some other molecules and atoms which are collectively called choline. Notice that phospholipids are amphipathic. What does that mean? It means that they have both hydrophobic and hydrophilic regions. There are some regions that love water and some regions that are water hating. So the hydrophilic head is actually charged. Well, water is charged too. So birds of a feather flock together. So charged atoms attract each other. So heads are hydrophilic and the tails are hydrophobic. Because of this, we know that membranes can easily form, and there's evidence that shows that lipids were present during early chemical evolution, that they can form under early Earth conditions. Lipids have actually been found on meteorites also. When amphipathic lipids are placed in water, they naturally form like little spheres. It's sort of like when you're looking at a dressing, an oil and vinegar dressing, and you see little globules of oil floating around in the vinegar and water. So this is the type of globules that I'm talking about that is forming the plasma membrane. So it can also form these things called micelles and the lipid bilayers like our plasma membrane. In figure 6.5, you see that lipid micelles are formed. Notice this is just a single layer, and the phosphate heads are facing out because they're hydrophilic, whereas the hydrophobic tails are facing inward. In B, you see the phospholipid bilayer, where the phosphate heads are facing out, and the hydrophobic tails are facing in. Phospholipids tend to form the latter, which is a phospholipid bilayer. In other words, you have 2 sheets of phospholipids, where the heads are facing out and the tails are facing in. The interesting fact is that these phospholipid bilayers form spontaneously. No outside energy is required to actually form the bilayer. So vesicles can be formed from phospholipids in the lab, and these vesicles can be used to test the movement of molecules across the membrane. So when we talk about artificial membranes, they're often referred to as liposomes. Here in figure 6.6, you can see some artificially made liposomes. In fact, don't they look like little globules of oil in a vinegar and oil dressing? So what are we testing when we look at artificial membranes? Well, we're testing permeability. What does permeability mean? It means the ability for molecules to move across the membrane. So some molecules may be able to move and others may not. But this membrane is separating two aqueous solutions. In figure 6.7, you see that there's a lipid bilayer, an artificial one that's placed in a beaker. And you see in part B that on one side of the membrane, you see that there are a lot of solutes or ions dissolved in the water. There's a high concentration, whereas the other side has no ions. So will these molecules or solutes cross the membrane? And the truth is some will, because membranes do show selective permeability. Selective permeability means that some molecules can cross, but others cannot. It just simply means that. You know what it reminds me of? It reminds me of a gated community. There's a lot of cars, but only some cars can go into the gated community. You know, those cars that have the code to get in and lift the actual barrier so that they can drive through. Well, the same thing is happening at the cellular level. It turns out that small or nonpolar molecules, that means those molecules that do not have a charge, can readily move across the phospholipid bilayer. If a molecule happens to be large, or it happens to have a charge, then it's going to move across slowly, if it is at all. So that makes sense, right? Because the largest part of the phospholipid bilayer are the fatty acid tails, and we know that those fatty acid tails have a neutral charge. I love this diagram. I love it. And you know I'll be asking you test questions based on this diagram on figure 6.8. What it shows is that small nonpolar molecules can readily cross a phospholipid membrane. But the minute that there's a charge, even though they're small, they're going to have a more difficult time crossing the membrane. And then as molecules get larger, like glucose or sucrose, they have a very hard time crossing the membrane, if at all. And even small ions like chlorine, potassium, and sodium have very low permeability. In other words, they can't cross the membrane, and the reason is because of the atomic charge. So what affects the permeability of a phospholipid bilayer? In other words, of a plasma membrane? What factors influence the behavior of the membrane? Well, it can be the number of double bonds, which makes it unsaturated and also decreases its density. It could be the length of the fatty acid tails, and you should be thinking about butter versus beeswax. Or it could be the number of cholesterol molecules that are embedded in the membrane. When I think of cholesterol, I think of the spacers that we often see in cement walkways, you know, that spongy double bonds or kinks in the hydrocarbon chains can actually increase permeability. So the more kinks there are, the greater the permeability is. The less kinks there are, the less permeability there is. So saturated hydrocarbon chains have fewer spaces, they have more stronger hydrophobic interactions, and they're denser and therefore less permeable. So more kinks, more permeability. It says here that hydrophobic interactions become stronger as saturated hydrocarbon tails increase in length. And I'm hoping that again that you're thinking about the beeswax. So the longer the tail, the harder it is to cross over the membrane. So longer hydrocarbon tails means less permeability. Shorter tails means greater permeability. I love figure 6.9 because it shows two things. It shows tail length and kinks. So if you look at the top, you see that the phospholipid bilayer has short tails, and you can see that there's double bonds because there's kink in the hydrocarbon tails. That kinks creates greater spacing, and with greater spacing, molecules can move more readily across the membrane or boundary layer. With an increase in tails and an increase in saturation, there's a decrease in permeability. I love this diagram. I hope you understand it thoroughly. Cholesterol can also reduce membrane permeability. So the more cholesterol that's embedded into the plasma membrane, the greater loss of permeability. In this experiment, they created a liposome. Remember, a liposome has an artificial membrane. And what they did is they looked at different levels of cholesterol and different temperatures. So if you look at the very bottom, you're going to see a graph. The x-axis represents temperature and the y-axis is permeability. Then you see three lines, and they're actually like three curves. The highest curve is in blue, which indicates the permeability when there is no cholesterol in the plasma membrane. So when you have no cholesterol, it increases permeability. The second line, which is in a mustard color, says 20% of lipids are with cholesterol. So notice the increase in cholesterol has reduced permeability. And the other line, represented in orange, has 50% cholesterol, and notice permeability is reduced again by 50 to 50%. So an increase in cholesterol reduces permeability. So what are the effects of temperature? It seems as if when temperature increases, what happens? Permeability increases, and that's why the lines curve to the top at the end of the graph. So it says here, how does temperature affect the fluidity and permeability of membranes? So it says here, phospholipid membranes are actually fluid. One thing that I always have to confess to my classes, that when I was learning about the cell and cell membrane, I actually used to think to myself that cell membranes were made out of like something akin to like plastic wrap or cellophane, that they were something more solid than just a drop of liquid or a drop of oil. But the truth is that plasma membranes are very fluid. In fact, they are like a drop of oil, and they're moving laterally. What does laterally mean? Side to side. Because remember, there's two layers. So in each layer, the phospholipids are moving side to side. They rarely ever flip from the top layer to the bottom layer. This diagram reiterates the idea that phospholipids move from side to side. There's lateral movement. Okay, so how do substances move across selectively permeable membranes like phospholipids? It's through diffusion and osmosis. Okay, so we already know that size matter. Small molecules can move more readily than large, and neutral or non-charged molecules can move more readily than charged molecules. So one type of movement is diffusion. So what is diffusion? Diffusion is the spontaneous movement of molecules from a high concentration to a low concentration moving across a membrane. So, in other words, molecules move with the concentration gradient, going from a high concentration to a low. When a concentration gradient exists, there is a net movement of molecules from that high concentration to a low. This movement is spontaneous, it increases entropy, and it does not require energy. And I hope you remember what entropy is. Entropy is disorder. So again, to actually take a look at this, diffusion is the natural movement of molecules from a high concentration to a low concentration moving across a membrane. Molecules are moving randomly, but there's a net movement moving down a concentration gradient. This is passive transport. And what does passive mean? It means that no energy is needed for molecules to move across the membrane along the concentration gradient. I love this example in figure 6.12. We see that there's two different kinds of molecules, those represented by purple triangles and then the white spheres. Notice that there's an artificial membrane in the middle of the beaker. So notice over time the triangles diffuse to the right hand side and the white spheres diffuse to the left hand side until there is still movement, but there is no net movement. So the concentration and equilibrium has been reached. What is osmosis? Osmosis is a special case of diffusion. In fact, it is the diffusion of water. So what would cause water to move? Well, you guys realize from doing the experiment in lab, the osmosis and diffusion lab, that sometimes molecules are too large to cross the membrane. So if they're too large, then the next best thing is water moving across the membrane to equalize the concentration. So water moves from regions of low solute concentration to regions of high solute concentration. Another easy way of saying that is water follows the solutes. Water follows the salts that are dissolved in water. In this example here, water moves across the membrane because the green molecules that are in the beaker are too large to move across the membrane. So water will move to the area of high solutes or high salts so that to equalize the concentration on both sides of the membrane. When we look at the type and the amount of molecules in aqueous solutions and we're comparing two, one on each side of the membrane, we are talking about tonicity. Tonicity, comparing the solute concentrations of aqueous solutions on both sides of a membrane. And we can have three situations: hypertonic, hypotonic, and isotonic solutions. The prefix hyper means above, the prefix hypo means below, and the prefix iso means about the same. In a hypertonic solution, water will move out of the cell and the cell will shrink, and if it's going to shrink, it's going to lose weight. In a hypotonic solution, water will move into the cells by osmosis. The cell will swell, and if water keeps on moving in, it can make the cell burst. In this case, the cell gains weight from the movement of water from the outside to the inside of the cell. And in the last situation, the solute concentration is the same on the outside and inside of the cell. Therefore, there's no net movement of water. The cell stays relatively the same size, and the weight relatively stays the same. I love the diagram in 6.14 because it shows the differences between hyper, hypo, and isotonic solution. So notice in a hypertonic solution that the solute concentration or the molecule concentration is much greater outside the cell than it is in the inside. And remember, water always follows the salts, or it follows the solute. So water will be sucked out from the inside of the cell, and therefore the cell will shrink and lose weight. In a hypotonic solution, there's more molecules inside of the cell than outside, and therefore water will flow into the cell following those solutes. And when it does, it'll increase in size and increase in weight. In an isotonic solution, the concentration of molecules is relatively the same outside and inside of the cell, and therefore there's no net movement of water, and there should be no change in cell size, nor should there be a change in cell weight. Membranes in the chemical evolution. I want to remind you from the nucleic acid chapter that the tipping point in chemical evolution was the evolution of nucleic acids, the information molecule. And if I want to even be more specific, more importantly, it was an RNA world first before it was a DNA world. And so remember, RNA can be a naked replicator. In other words, it's a ribozyme. So what's missing to make it a cell? Well, the missing part is the membrane, which is made of the phospholipid bilayer. So if there's a membrane, then RNA is no longer a naked replicator. This is the prototype for the first cell. In this figure 6.15, you see that you have nucleic acids inside a membrane, and this membrane is made of fatty acids or lipids. So this is a protocell. This is the beginning of cell formation and life itself. The last subject that's in this chapter is how proteins can alter membrane structure and function. Well, proteins do play an important role in the phospholipid bilayer, which makes our plasma membrane. So plasma membranes contain proteins. So what is their function? To me, I think that their function is doorways. They're doorways into the cell. They're doorways for those molecules that are too large or they're charged and cannot move across the phospholipid membrane. Proteins, just like the phospholipid bilayer, are amphipathic. In other words, one side is polar and the other side could be nonpolar. And it folds into shapes to form different kinds of gates and channels. So here you see in figure 6.16 that some of the amino acids making up this protein are green and then yellow. This is indicating polar and nonpolar regions, or charged and non-charged regions. And you can see how it readily fits into the phospholipid bilayer in part B of this diagram. This leads us to the idea of a fluid mosaic model. Well, what does fluid mean? Fluid means like a liquidy movement of molecules. And we already know that there's a lateral movement of phospholipids. So what does the mosaic mean? The mosaic, like when I think of mosaics, I think of those fancy tile pictures that you find in Italy, in the Mediterranean area. Well, that means that they're using different colored tiles, or they're using different colored rocks to make a scene. Well, that's what's happening at the cellular level. Now it's not just phospholipids, but all different kinds of proteins are embedded into the phospholipid bilayer, making it look like a mosaic. So it's the fluid mosaic model. Now, in the early days, like the 1950s when I was born, they didn't know exactly the relationship between the phospholipids and the proteins. And one idea was the sandwich model, which we know now is not is not exactly what's happening. In the sandwich model, the protein is sandwiching the phospholipid bilayer. And in fact, this turned out to be proven false. So the correct model is B, the fluid mosaic model. So you see all different kinds of proteins embedded in the phospholipid membrane, and notice they cross the entirety of the phospholipid membrane. When they cross from the outside to the inside, they can be called transmembrane proteins, or they can become integral proteins. So either name, integral or transmembrane, mean that proteins cross all the way through the phospholipid membrane. Notice there are also proteins that just stay on one side. They either stay on the outside or the inside of the membrane, and these are called peripheral proteins. In this experiment here, this experiment shows that many proteins are transmembrane proteins. So how did they do it? They freeze-dried the plasma membrane, and then they separated the phospholipid bilayers. And when they did, they actually exposed these pits. Well, what formed the pits? Well, the pits were formed by the protein that should be embedded and going all the way through one layer to the second one. And this in fact reveals or supports the idea of the fluid mosaic model. Again, when proteins span the membrane, that means when they go from the outside to the inside, we can say that they're integral proteins or transmembrane proteins. Peripheral proteins either stay on the outside or they stay on the inside, but they definitely don't go all the way through the membrane, not like integral or transmembrane proteins. Okay, so they've done some studies to actually study proteins to see what their function is while they're embedded into the phospholipid bilayer. So they've separated these proteins using detergents. And what they found was that these proteins could be considered channels, carriers, or pumps. In other words, they're special doorways into the cell. And here's an experiment where you can separate a protein from the rest of the phospholipid bilayer using detergents. Okay, so the first type of protein that you can find in a phospholipid bilayer is an ion channel. Well, what's an ion channel? It's like a tube, it's like an opening, it's like a doorway leading into a room, and it allows for molecules to go in and out. So specific ions can diffuse through or they can diffuse out. So this is important for ions because ions have a charge. And because of that charge, it's very difficult for them to cross the fatty acid tails in the phospholipid bilayer. So how do they move across without any energy? Well, they form this electrochemical gradient. Now, we've talked about chemical gradients, but we've never talked about an electrochemical gradient. So what's the difference? Well, if we look at sodium, sodium is a chemical, but it also has a positive charge. So if sodium accumulates on one side of a membrane, you have an increase in sodium, but you also have a big increase in positive charges. And that will be the driving force to drive sodium across the membrane from a high concentration gradient to a low. So because there's a charge involved, it's an electrochemical gradient. In figure 6.20, it shows exactly what I was discussing. Notice all of these sodium ions accumulating on one side of the membrane. Well, if there's an ion, an opening like a doorway to the inside of the cell, sodium ions will readily cross the phospholipid bilayer because there's an electrochemical gradient. So you're going from a high concentration gradient to a low concentration gradient. And we're doing this through a protein doorway. In this example here, they talk about a specific channel. And this one is for the movement of chloride. So chloride is also an ion. It has a negative charge. And notice if this protein is missing, and is not embedded in the plasma membrane, then chlorine cannot move freely across the membrane. And this one little ion channel leads to cystic fibrosis, that is a genetic disorder found in people of European descent. Because there's no free movement of chlorine, mucus can form around the organs of human beings, and it can often lead to early death. Channel proteins are selective, so each protein is meant for the movement of one ion or one particular type of molecule. So each channel protein permits only a particular type of ion or small molecule to pass. One of the most famous channel proteins are aquaporins. They allow water to move across the plasma membrane, because keep in mind, water is charged. Here's an example of an aquaporin. Notice that water is moving across the plasma membrane, and an aquaporin only allows water to move across the membrane. Another kind of protein that's embedded in the plasma membrane is a gated channel. So it's a doorway, but this time there's like a gate or a door, so it can open and close. And what usually affects the opening and closing of this door is a change in the electrical voltage. You know, like maybe a charged molecule moving across the membrane, it changes the electrical voltage and it will either open or close the gated channel. I love this example in 6.23. Notice that you have plus charges on one side of the membrane and negative on the other. And in the second diagram on the right-hand side, notice that the charges have been reversed. Now it's negative on one side of the membrane and positive on the other. This reversal in voltage has opened up the gate to the protein, allowing molecules to move. There's another type of protein called a carrier protein, and this carrier protein helps with facilitated diffusion. So that's a good test question. What's the difference between diffusion and facilitated diffusion? Well, if it's diffusion, we know it's passive movement. We know it doesn't require energy. And why is that? Because molecules are moving with the concentration gradient. In other words, they're moving from a very high concentration to a low concentration. They're following the concentration gradient. So what does facilitated diffusion mean? It means that there's a helper, and the helper is the protein itself. So it is still a no energy activity or movement of molecules, but it's facilitated by the presence of an integral or transmembrane protein. So in facilitated diffusion, you can have carrier proteins that actually change shape to transport molecules across the membrane. One of the most famous carrier proteins is GLUT1, and GLUT1 carries glucose from one side of the membrane across to the other side. So GLUT1, what it does is it recognizes glucose, and only glucose can fit into the membrane, and it moves it from the outside of the membrane to the inside of the cell. In this diagram, you can see that this carrier protein is really fancy, and the protein itself moves. It opens out, closes, and then opens inward, releasing glucose. So this carrier protein is meant for the movement of sugar or glucose across the phospholipid bilayer. Another kind of movement of molecules is active transport. Active transport needs ATP. They both begin with the letter A, so that should help you to remember active transport requires energy in the form of ATP. And the reason for this is because molecules are moving against the concentration gradient. It would be like me pedaling up to the top of the Franklin Mountains. Oh boy, howdy, that's going to take a lot of energy. Another example would be is say there's a fancy club that opened downtown and it's already full of people and there's a long line of people trying to get in. So the odds of you getting in are very, very small unless you get help from the bouncer. The bouncer is ATP, active transport. That means you're moving against a concentration gradient. You're moving from a low to a high concentration gradient. You're moving in the opposite direction. So for active transport, how is that possible? How can you move against a concentration gradient? Well, one of the ways is through a pump. And one of the famous protein pumps of all time is the sodium-potassium pump. And the reason why this pump is important is because salts affect our circulatory system and our heart health. So the sodium-potassium pump is a very important pump, and it's an example of active transport. In other words, ATP is needed for the pump to work. So both sodium and potassium are transported against their concentration gradients. In this diagram here, you're going to see that sodium is being pumped out of the cell out into the outside, which has a high sodium concentration. And on the other hand, potassium is being pumped into the cell, and it too is being moved against its concentration gradient. So when you're moving against a concentration gradient, it requires energy. This is called active transport that uses ATP. There's also such a thing called secondary active transport. And what active secondary transport is that other molecules can move with one molecule that's moving against its concentration gradient. So in other words, it's sort of piggybacking on the movement of other molecules. It's good for you to know, but this will not be on the test. This will not be on the test. So let's go over and summarize what we've just gone over. There's passive transport and active transport when we talk about molecules moving across the phospholipid membrane. So passive transport includes diffusion and facilitated diffusion. So both mean that molecules are moving from a high concentration to a low concentration following a concentration gradient and moving across a membrane. Facilitated means it's facilitated with ion channels, gated channels, and carrier proteins. All of these are helping or facilitating movement across. Then the other form of transport is active transport, and this is where energy is needed for molecules to move across to move against their concentration gradient. I love this diagram because it shows the difference between diffusion and facilitated diffusion, and then again it shows the difference between facilitated diffusion and active transport. And in fact, this frame on active transport is the best one in the entire chapter. I hope you've understood this chapter. Best of luck.